Hello, everyone. It's my pleasure to be here with you the last. So I hope that you still have some of your votes left. My name is Seifelden. I'm a PhD student at the Polytechnic University of Madrid. And I'm here to share the latest research from our lab. We have been recently working on an R package called MATE-R that optimizes the crossing block stage in breeding programs. We use new equations that are general, and it has or supports different metrics to control genetic diversity with. But not only that, we also optimize another critical stage in breeding programs that has been neglected by many, well, up until now. So let's get into it. Our lab is in the Center for Plant Biotechnology and Genomics in Madrid, and we are mainly focused on improving the breeding methodology in case you would like to visit us. My talk will be divided into the following 5 sections. So I will start by giving a background on what kind of problems do we have and the derivation of one of the new metrics that we developed for controlling genetic diversity, and I will go through how did we evaluate it using stochastic simulations. So here we are looking at the big picture of plant breeding, where we start by crossing elite parental lines to generate new progenies that will be evaluated across different years and trials until we obtain the new ones that will be recycled into the parental pool. The problem here is that during the crossing and selection, we always want to select the best, and we also want to cross the best with the best. However, these best individuals are highly correlated genetically in terms of, in a sense, that they have or share lots of genes that enable them to confer the high performance. And focusing only on these individuals is extremely detrimental to the variance component of the breeder's equation, which drives response to selection, which is further exacerbated in genomic selection programs compared to phenotypic selection ones. So how do we solve this? Optimal contribution selection was proposed as a concept pretty early in 1958, and to select the elite parental lines based on their contribution to the next generation. However, it has limitation that it assumes random mating, and we know that that's not true in real breeding programs because we are crossing the elite by elite parents. So instead, optimal mate allocation was developed to overcome that limitation, or as we would like to refer to it, optimal genomic mating, where we are trying to find an optimal mating plan that balances genetic gain with variability under realistic constraints, practical constraints. So, of course, optimal genomic mating is not a new concept. It has witnessed lots of methodological advances across the last 15 years, starting from Kinghorn, who first solved the problem using evolutionary algorithms. And most notably, you will find genomic mating, alpha mate, simple mating, and most recently, convex OMA, and our package MATE-R that you could find it to use here in this link. We have currently submitted the manuscript to explain everything. So in optimal genomic mating, we are trying to find a mating plan that balance 3 factors. The family mean with 2 sources of variability in breeding populations, within and between family variability. Let's see together how do we compute them in our MATE-R framework. So first, you can have some families, just like this red one right here, which has higher mean than the green family. However, you could argue that the green family is much more useful because it has bigger variance. So it will allow us to capture some superior individuals right here if you are able to exploit it. At least that's what we hope. Which takes us to the concept of usefulness criterion, which is the linear combination of family mean and within family standard deviation. It also takes into account within family selection accuracy and intensity. So we compute or estimate with a family mean using different equations. We start to compute the genotypic effect, considering additive and dominance, and also the genotypic frequencies of the progeny in a single loci, and then we propagate it across the entire genome to obtain our family mean. And it works for diploids, allopolyploids, and also autotetraploids. These equations are arguably much more flexible compared to Falconer equations, because Falconer equations usually predict for the F1 progeny. However, these ones you could predict after any number of selfing cycles. So, and for the within family standard deviation, we need two pieces of information: family correlation matrix that can be approximated from the LD information of the parents, and also a vector of single loci additive plus dominance standard deviations to take into account the covariance structure between additive and dominance effects. So far we have talked about the family mean and within family variance, but how about between family variability, which is what we really want to control? Usually between family variability is formulated in terms of inbreeding. And inbreeding is a very complex concept with lots of definitions. Is it a correlation as described by Wright, or a probability as Malikot said? Should we estimate it using pedigree or genomic relationship information? Even if it's more appropriate to estimate it using pedigrees, we usually don't have high quality ones in our data. So here you could see that we have done a very small simulation. We have computed the inbreeding values or average group ancestry across 30 years for a running line breeding program. And these are the inbreeding rates. You can see that when they are estimated from the numerator relationship matrix, it's not the same estimates that can be obtained from a G matrix. They are quite different, even though they could be correlated. So to overcome all of these problems, our motivation was to develop a new metric, which is called the proportion of additive standard deviation lost, or ProbSD for short. And this metric is interpretable because we know that from the previous equation, the response of selection depends on the additive standard deviation. Now let's see together how did we derive that metric. So let's say we have a parental pool and a mating plan that we are interested to implement. Their genomic estimated additive values are coming from a distribution with mean 0 and the variance of the genetic relationship matrix. So due to selection, we expect that the additive standard deviation of the mating plan is going to be lower than that of the parental pool. So the proportion of standard deviation lost can be computed that way. Of course, these values are unknown. However, we can estimate them using the expected standard deviation of an infinite amount of samples from their multivariate distribution. And for that, we need two things. First, the expectation of the sample variance that can be computed this way, following Surley and Endelman, with the addition of a correction factor, because we need the sample variance, not the population variance. And then we need the variance of the sample variance, which is, you could call it the standard error variance. To compute it, you could follow our latest publication from the lab. And it could be very tempting to do it that way, that you take the square root of the expected sample variance. However, this is incorrect because the square root of the expectation is not equal to the expectation of the square root, which is what we really want. Luckily, we do know that the sample variance is coming from a gamma distribution with the following shape and scale parameters that could be used to obtain the expected standard deviations that we want for both the mating plan and also the parental pool to compute or estimate the proportion of additive standard deviation lost. This metric is interesting because what you predict or want is what you get. So you could see here almost a perfect correlation between realized and predicted ProbSD values in diploid and autotetraploid crops. However, this is under random mating of parents, which we know it's not true. Under selection, this will follow closely the depletion of the genic standard deviation. Now let's see together how did we evaluate our method. Of course, we used AlphaSimR to perform stochastic simulations for phenotypic and genomic selection schemes, a typical line breeding program. We simulated the exact same program that's published in these two publications. Please refer to them for more details. I'm not going to go through them all. And that's to make sure that we are not choosing biased parameters to test our strategies or metric. And we have done the same for a line breeding program as well. So here you have our simulated scenarios. We have two control ones with random mating, so phenotypic and genomic selection, that takes into account no factor at all during mating. And in contrary, we have truncation, genomic mating, or cross-selection scenarios, where we select the families based on the mean and the usefulness criterion, where we take into account family mean, and for the usefulness criterion, we have family mean, and within family standard deviation. And finally, optimal genomic mating using our MateR package, where we take into account all three factors together to balance with diversity. So let's get into the results. But first, I have to refresh for you the concept of Pareto frontier. So we are trying to balance two conflicting objectives, right? So this blue strategy right here is very bad because it saves a lot of variance, but it doesn't deliver gain at all. And the red one here is also equally bad because it delivers lots of gain, but at the expense of depleting all variability in the breeding population. We typically want something that's very close to that theoretical purple one right here, which is, of course, non-existent. And that's why we would obtain normally a Pareto front of solutions like that, and we should look for a solution that is as close as possible to this purple strategy. We have done the same, simulating the different scenarios. And you could see here the trade-off in the final year, where genomic selection with random mating was suboptimal for gain, but it was very good for diversity. And in contrast, the two truncation mating scenarios achieved relatively higher gains at the expense of depleting more variance. And in the middle, you could see the different solutions with probes D values ranging from 0.5 to 10%, with 0.5% probes D being the most diversity strict. We have done the same for hybrid breeding, and you could see that the optimal solution is around 2 or 3% here, or you could argue that it's 0.5 if you really care about variance. So far, we have optimized the crossing scheme, but how about recycling, which is neglected? Normally, we are recycling the top-performing individuals using truncation recycling, but what we should really be trying to do is to recycle, to find those individuals that balance gain with variance. Here are, if you remember, the Pareto solutions for both programs, where we used only optimal genomic mating, and at 3% was the optimal solution, ending at 0.25 and 11 units of variance. When we combine optimal recycling with genomic mating, you could see much more interesting solutions right now that resulted in the expansion of the Pareto front, and the optimal solution would be somewhere around 4 to 5% probability, which is essentially saving double the amount of variability that was saved by optimal genomic mating alone, without compromising gain relative to the truncation mating scenarios. And if you are going to compare it to the truncation mating scenarios, you will see that it saved almost 4 times or fold the amount of diversity. To show these results, I'm going to go quickly. I'm about to finish. So we can regress the achieved standardized gain on lost genic standard deviation, and the slope for that regression line would be equal to the conversion efficiency of variance into gain. We have looked into our results, and you can see that phenotypic selection with random mating has the highest conversion efficiency. It's really difficult to beat compared to the two truncation mating scenarios used as a reference here. And you can see that optimal genomic mating provides comparable solutions with random mating genomic selection. And finally, the integrated strategy of optimal recycling and genomic mating, it achieves conversion efficiencies that are getting closer to the phenotypic selection program with random mating. And that was consistent in the other program as well. Finally, to sum up, we propose probability as an intuitive metric that could be used for different breeding schemes and populations. And we showed that simple modifications in a single stage in breeding schemes that are critical could make a huge difference to achieve genomic selection programs that are sustainable. And thanks for your attention.